topic is about nuclear pore complex NPCs. Nuclear envelope is perforated with thousands of pores. Each is constructed with from multiple copies of several dozen different proteins called nucleoporins. The entire assembly forms an aqueous channel connecting the cytosol with the interior of nucleus nucleoplasm. When the material are to be transported through the pores, it opens up from one channel some 27 to 41 nanometer wide, large enough to get such assemblies as is ribosomal subunit through. Transport through the nuclear pore complexes is active. It that is require energy. Many different carrier molecules such as specialized to transport particular cargo. Docking molecule in the NPCs represented here as colored rod and dixies. In this, energy is used because it's an active transport in which energy is required. The next is import in the nucleus into the nucleus. Proteins are synthesized in the cytosol, and those needed by the nucleus must be imported into it through the NPCs. They include all the histone needed to make the nucleosomes, all the ribosomal proteins needed for the assembly of ribosomes, all the transcription factor is, for example, steroid receptor needed to turn genes on, all the splicing factor needed for processes and pre-messenger RNA into mature messenger RNA, that is to cut out intron region and splice the exon regions. Probably all of these proteins has a characteristic sequence of amino acid called a nuclear decolonization sequence that target them for entry. Next is export from the nucleus. Molecules and macromolecules assemblies exported from the nucleus, including the ribosomal subunit containing both ribosomal and proteins, ribosomal RNA and proteins, messenger RNA, mRNA molecules accompanied by the proteins. Next is transfer RNA molecules also accompanied by proteins. The last is transcription factors that are returned to the cytosol and await reuse. Both RNA and protein molecules contains a characteristic nucleus export sequence NAC needed to ensure that their association with the right carrier molecule to take out to the cytosol region. The next is nucleosomes and DNA replications. As is the case of transcription, the DNA legs must open to allow DNA replication to proceed. This too requires that the nucleosomes preceding the replication fork to be removed and then quickly assembled as the leading and lagging strands are synthesized. The next is nucleolus. During the period of cell division, when the ribosomes When the chromosomes are in their extended state, one or more than 10 in human cell have loop extending into spherical mass called nucleus. Nucleolus here are the synthesized three of four of the four kind of ribosomal molecules 28S, 18S, and 5.8S used in the assembly of large and small subunit of ribosomes. 28S, 18S, and 5.8S ribosomal RNA is sub-transcribed by ribosomal RNA polymerase so that in the yeast the 5S ribosomal RNA molecules as well as the as well as transfer RNA molecules are also synthesized by ribosomal RNA polymerase 3 in the nucleus. Once formed, ribosomal RNA molecules associate with the dozen of different ribosomal proteins used in the assembly of large molecules or large and small subunit of the biosomes. The transport through the nuclear pore complexes is active that is required energy